Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Steve Sando was peering through a microscope at a minuscule worm squirming to escape a light when he made a surprising discovery. The type of worm he was observing, Caenorhabditis elegans, uses a muscular pump to swallow up tasty microbes from its surroundings. But when a worm was exposed to light, Sando noticed one day in early 2014, that suction reversed course, jetting liquid out of the worm's tiny, transparent throat. As he watched the little creature make this movement, the first thought that came to mind was, "Oh my God, it must be spitting," recalls Sando, then a doctoral student working under MIT molecular geneticist and neurobiologist Robert Horvitz. "I pulled my laptop out of the microscope and ran down the hall to show everyone in the lab. Before this, the model was that the smallest controllable unit of muscles is a single muscle cell. By recording several hundred worms and carefully sifting through countless hours of footage, Sando and his colleagues revealed that individual muscle cells in the C. elegans mouth are able to carry out two tasks simultaneously by contracting in different patterns at different ends of the cell. Before this, the model was that the smallest controllable unit of muscles is a single muscle cell." Sando says, "So this finding changes how we think about how animals generate behavior." The work that set the stage for Sando's study had been initiated several years earlier by another former GRA. Curiously, he discovered, light not only changed how the worms moved, but also made them stop eating. He also found that the proteins controlling the worm's ability to evade light were encoded by genes related to sequences encoding taste receptors in insects. Through a series of experiments, Botla revealed that these genes also controlled the worm's response to bad smells, specifically, to the odor of hydrogen peroxide, which is generated when light hits biological tissue. Following this movement, the worms would occasionally blow bubbles out of their tube-like mouths. Andrei Krauss-Sando says he wanted to get a closer look at this strange behavior. He decided to slightly flatten the worms before viewing how they moved in response to light. The thinking was that, if we squish them a little bit, maybe that'll slow them down and help us see what the muscles are doing, he says. This adjustment did the trick. It worked so well, in fact, that Sando pinpointed muscle motions that appeared to be spitting behavior in the first batch of flattened worms he observed under a microscope. After confirming that this behavior was indeed spitting, using tiny plastic beads to show that liquid was being expelled from the worms' mouths, Sando and his colleagues published that initial finding in 2015. Typically, when a worm eats, 3 muscle cells within the pharynx contract and relax rapidly to propel food into the body. The researchers found that when the worms spat, the front portion of each muscle cell contracted, holding the mouth open, while the back portion continued a pumping motion, expelling food from the worm's mouth. The finding of a compartmentalized signal in a muscle cell was completely unexpected to me, says Manuel Zimmer, a neurobiologist at the University of Vienna who was not involved in the work. Whether or not this phenomenon of different, simultaneous actions by a single muscle cell exists in other, larger animals or is unique to C. elegans remains an open question. Broadly speaking, however, this study is a significant step toward understanding how neuronal circuits work, Samuel adds. To date, there has yet to be a complete neurophysiological model of even simple behaviors, he says, but these guys have a big piece of that puzzle with this, and it's a step toward real models of real brains. Quote, "Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you."